You might be getting tired a little bit. And today is a great pleasure for me to present my old friend that I've known since school and who since then became a prominent scientist in her field, so Julija Krupič. She graduated from Faculty of Physics here at Vilnius University and then moved to London. First, she received Master of Research in Imaging Science at Imperial College London. And then she moved to work with Professor John O'Keefe. John O'Keefe is a Nobel Prize laureate in medicine in 2014, and Julija worked with him to receive her PhD in neuroscience in University College London. Thereafter, Julija received funding from Sir Henry Wellcome Fellowship and stayed there to start her independent line of research. Her scientific data was and is still presented in publications in very high impact journals such as Science and Nature. And recently she has moved to University of Cambridge to become independent group leader there. She's a lecturer at university and Sir Henry Dale Fellow, and she's establishing now her own group. Julia's research is funded by the Wellcome Trust, the Royal Society, Isaac Newton Trust, Kavli Foundation. And her aim is to understand the cellular basis of our internal GPS. So we'll probably have to know a bit more about that after your talk. Dr. Julija Krupič.
Thank you very much. Can you hear me now? Okay, so thank you very much, first of all, to organizers for kindly inviting me here. It's a great pleasure to be back home in Vilnius University, one of the greatest universities in Europe, in my opinion. As Urte said, I studied physics here, but then switched a little bit my focus to applying, I would say, still quite widely physical sciences to investigate and research biological problems. And in particular, a system called Hippocampus, located here in our temporal lobe. So this is a human brain skull. I'm not working with humans, so you will see that the majority of my own work will be done and will be presented in rats. Other rodents as well, but in this presentation, rats. So you see hippocampus, temporal lobes, hippocampus is here. It's a structure which was, when John O'Keefe and Edward and May Britt Mosers were awarded Nobel Prize, was coined as a GPS, so internal map of our brain. So if you look at the connectivity, this is like a structural scheme of connectivity coming from the retina to the LGN, visual cortex, and other higher visual areas. You can see how the signal, right from the sensory modalities, so right from registering photons from the outside, goes all the way to the hippocampal formation. So this is that region, located pretty much very, very deep from a sensory modality. So in this case, you see the hierarchical representation of the visual system, so visual cortex and so on, but actually that would apply pretty much to any sensory modalities in our brain, so auditory cortex, tactile, and so on and so forth. So it's a system, it's a brain structure which receives a very rich amount of information about the external world, also about the internal world as well, so inputs from brain regions related to emotional states and other internal states, integrates that information to create so-called maps, so spatial perception of environments. And we are interested to study how it does it and how then these representations are used to perceive space, navigate, remember events, and so on and so forth. So the major focus or interest in the hippocampus started pretty much in the 50s, and especially in the context of memory. So its role in memory representations, and a particular type of memory called episodic memory. So we have well, roughly speaking, two types of memories, procedural and declarative, which episodic is one of them, where essentially it's in our ability to form representations of events happened to us at some point. And here you see this person called, at that time he was called H.M., but his real name is Henry Mollesen. And he was a patient who suffered from very severe epileptic seizures. And at the time to deal with it, this very prominent neurosurgeon, William Scoville, and his colleague, Brenda Milner, decided to perform very pioneering surgery on this person by essentially removing a chunk of his brain, a lot of his temporal lobes from both sides, which also included very much hippocampus, intrinal cortex that you will hear about, and some other structures as well. And so what happened then after surgery is that indeed his epileptic seizures were somewhat more manageable when he woke up. But what also they noticed, Brenda Milner in particular, which she was awarded a Kavli Prize for that, was that he was not able to form episodic memories anymore. He was not able to show any type of learning of new events happening to him. So for example, if you meet him, he will greet you, you will have a conversation, you will leave a room, come back, and he will treat you as if you just came in, he just has met you, and so on and so forth, and he will more or less have the same or similar type of conversation again and again and again and again. So he kind of relived a permanent present tense, if you like, which was very, very nicely described in Susan Corkin's book, who worked with him a lot. And that subsequently, there were more patients which, for one reason or another, lost the hippocampi and showed this profound anterograde amnesia and partial retrograde amnesia, which means that they also lost some knowledge about events recently happening to them. For instance, he could remember some events from his childhood, or allegedly he could. It's very difficult to check that for sure. Which means that, you know, some episodic memory probably is already in the cortex, but at least the recent one and the one that we are learning as we go along takes place in the hippocampus. So here you can see this is a normal brain. This is a cut through that brain in this direction. And you can see that in HM patient, a large chunk of temporal lobe is just completely removed from both sides. Okay, so that's was very clear that the hippocampus plays absolutely crucial role in episodic memory in humans. But what also was discovered, actually in rodents first, was that it's absolutely a key structure for spatial navigation. And in particular type of spatial navigation, because of course we can navigate using different strategies. So for example, I can navigate to that poster because I see it, so I'll just walk right to it. That's not hippocampal dependent. Or we can have navigation based on us remembering different turns, for example. That's also not hippocampal-based navigation. But if we're talking about the most flexible form of navigation, where we're kind of freely exploring environment, building a kind of map, internal representation of that environment, that is hippocampal dependent, which essentially allows us to make shortcuts, detours. So it allows us to take trajectories which we haven't visited before, just because we built a kind of a map, subsample maps, where we know roughly a relationship between different places in 2D or even 3D space. So it became very clear that hippocampus is very important or crucial for that role as well. And you can immediately probably see how that can have a very relevant impact to something like episodic memory, which is very likely to be mapped on a kind of spatial domain. If you think about it, you remember some events happening to you, they usually take place in space. So they probably related like that. So what happened in humans after it became clear that it is a spatial map, cognitive spatial map of the brain in rodents, which will come back or come forward in a minute. The way it was checked in humans was using virtual reality in 1998 or so. So that was the time when reality games just became available. So what happened, scientists essentially took one of the engines, reverse engineered it, if you like, removed all the bad characters, guns and whatnot, and left just quite a large, reasonably large area, part of the city, let's say Old Town, downtown, where people could just freely walk, explore streets, cinemas, play pool, whatnot. So they were asked to explore this environment to kind of build an internal representation of this environment. And then they were asked to navigate from any starting location to any other location. So as you can see here, so 3 paths to the goal. So starting location, let's say, is in this place in A, and then finishing location is in B. And 3 different individuals are deciding what would be the best way to go from A to B. So you can see that one of them just wandered completely in the opposite direction, so had not a very good internal map, let's say, of that city. The other one probably used something which was not hippocampal dependent or did not have the optimal route to it, where it basically probably knew how to get from this place to B quite easily, but then didn't quite know how to connect A and B without taking this detour. And finally, a third person just could go straight from A to B, getting the most direct route. And what happened is that for this study, they used a technique called fMRI, which essentially approximate neural activity measuring the blood flow in the brain. And what they saw is that the more accurate the decisions were in terms of navigating to the goal in the most direct route, the more active hippocampus was. So that was a kind of a link already telling that hippocampus is important for navigation in humans as well. Another indication was followed up in this follow-up study done some years later, which became famously called a London cab driver study. And of course, London cab drivers, at least used to be, so now with the automatic GPS navigation system, perhaps it's somewhat lost, but at least used to be the best probably navigators in the world because the streets are quite complicated. And in order to pass the exam to become a cab driver, people had to demonstrate a really profound knowledge getting from one small street in London to another street. And so when people, cab drivers, were asked to drive from one location somewhere else in London, and the hippocampi volumes were measured, again, it became clear that actually they had increased hippocampi. And then when that was compared with, let's say, bus drivers who also drive in London, so it's equally stressful situation, perhaps the time spent was also controlled for, and so on and so forth, they didn't show that trend. So again, it had something to do with this flexibility, the need to use a map constantly or hippocampal activity constantly to navigate in these streets. So what we were interested in is to look at the cell activity, neural activity in the hippocampi, but on a kind of a system level basis where we could characterize the activity of individual neurons as well as the ensembles of neurons while animal navigating, trying to understand how exactly they compute these functions. So here you can see human brain again, and the hippocampus shown in yellow, and this is a rodent brain. So for the purpose of this talk, it's a rat. It doesn't have to be, it could be a mouse. They're quite similar in many respects, at least. And you can see that actually in a rat hippocampus shown here in yellow is actually very large brain structure compared to other brain structures, and compared to relation between the hippocampus volume and the rest of the cortex for humans. And if you also looked at the kind of, you remember that diagram that I showed you before, how hippocampus connects to other brain regions and so on and so forth. So it's all, again, very much preserved between a rat and a human, for example. So it's a very good system in that sense to look at the single cell activity and network activity to understand how hippocampus does what it does. So here, and before that, I mostly stress hippocampus, but actually my own work so far mostly concentrated on another area shown here in green, called medial entorhinal cortex, which is one of the major inputs and outputs from the hippocampus to neocortex. So again, if you remember that diagram that I showed you, and the hippocampus was a gem, a diamond on the crown, right just below it, it was entorhinal cortex, which was a kind of interface to all the rest of the brain regions. And you will see why that is also very, very important for representations that we find in the hippocampus. So this is the cut through the hippocampus. You can see that unlike neocortical areas, which have 6 layers of cells, it's actually very distinct. It's a 3-layer so-called structure. So see one region shown here, and dentate gyrus, which I'll talk a little bit in a minute, shown here as these kind of blades in different colors, just because we have these very fancy techniques where we can image individual neurons in individual colors. Hence, we can, in principle, characterize the activity and they relate to connectivity, genetic properties, and so on and so forth. I'm not going to talk about it in this talk. Okay, so cells and the hippocampal formation and entorhinal cortex. So I'm slowly drifting to the entorhinal cortex. So the major cell types in the hippocampus itself are so-called place cells. So what you see in here is the firing properties of 32 different place cells recorded in a square environment. And what it essentially means is that warm colors in red represent where that particular neuron, so one neuron, was active most. And blue colors, so cold colors, represent where the neuron was silent. So it means that essentially the animal was walking in this square environment, and that particular cell was more or less silent everywhere except for this corner. And this cell, again, in this very similar corner and so on. But you can see very quickly that even with these 32 cells, and in the environment, which is, I think in this case, it was something like 60 by 60 centimeters, it's not large, but still 32 cells more or less made an entire map, and it's also a redundant map in a way, of that environment. And there are hundreds of thousands of cells in the hippocampus, and we think, based on the experimental evidence, that the majority of them are either place cells or somewhat related to place cells. So you can see it's a huge capacity to encode different environments. And what's important about this system is that when animal leaves, let's say from this square to another square in a different room, different context, and so on and so forth, these cells will behave in a way that in the field we call it remap, so global remapping, which means that they will change their preferred firing locations in what now seems to be completely unpredictable way, meaning that there would be no consistent relationship, let's say, between this cell and that cell, which were more or less overlapping in this environment, but they completely can change the firing locations, or even one of them can become silent, and so on and so forth. So that's also quite important to remember, perhaps, or to note, is that they are reused in different environments. It's not like one cell is reserved for one place. That's not how it works. So these are place cells. And then these three guys are other three spatial cell types, which actually are not located in the hippocampus itself, but upstream, or whatever you want to call it, the downstream, upstream, so interface area between the hippocampus and neocortex, called medial entorhinal cortex, so the area that I mentioned to you previously and the area that I personally worked most. So let's start with let's say, the simplest cells, let's call them, not to insult anybody, but so boundary cells, they are essentially neurons in this medial entorhinal cortex, which are active close to the boundaries of enclosures, so close to the walls. So in this case, it's an example of one cell active in the north, but actually you have south, west, east, combination of any of these, and so on and so forth. Then we have head direction cells, which showed in a polar plot, which means that these cells are active whenever an animal is facing in a particular direction in allocentric frame of reference, so in a room frame of reference, not in the egocentric, so not in respect to the neck or anything like that, but actually the absolute one, somewhat like a compass of environment. And finally, grid cells, which are just like place cells, they're actually active in a discrete spatial portions of the environment, but in their case, these fields, so grid fields, well, first of all, it's multiple, a number of them, so a whole lot of them, and they are distributed not in some kind of random way, but they are distributed in so-called hexagonal symmetry, so really like a crystal of the brain, if you like, these cells. And that's where my major focus was, and that's where I will concentrate for the purpose of this lecture. So remember when I told you that place cells do remap when we go from one environment to the next, and so on? So what's special about these entorhinal cells in general, and grid cells included, is that they do not remap. So if the animal is not disoriented or anything like that, so can keep a track of directionality and so on, you can very easily predict how border cell or head direction cells or even grid cell, in majority of cases, I'll show you where it's not the case, will look like in a different environment. So, if animal is not reoriented, so north is north, so the head direction cells will keep that direction, and south is south, and so on. Borders stick to the north as well. And grid cell, very much so, will preserve its orientation, the scale, and so on and so forth. So that invariance and periodicity prompted, just after the discovery, so which happened in 2005, but just immediately after, suggestions and working hypotheses that they actually act as a ruler of the brain. So it's a system for measuring essentially distances. So now you can see how we have this very nice navigational gear, if you like. We have cells, compass cells, we have border cells. They are maybe perhaps not necessary, but they are there, if you like. And we have a ruler, and then we have a place representation. And then various theories emerge how these grid cell, so crystal-like structure could be formed in the brain, because I must emphasize that this crystal structure is not so much in anatomical space, it's actually a self-firing property, which is absolutely remarkable and was awarded a Nobel Prize very, very soon after its discovery, actually, so only 9 years after its discovery. And so there were two major classes of models, computational models, trying to explain grid cell structure. So one of them was continuous attractor network model, which in a nutshell essentially postulated that the grid cell response properties emerge in the entorhinal cortex itself, and that firing property emerge as a network property of these cells, if you like, the way they are connected, without going into details of that. And oscillatory interference model was very different in a way that it used the other properties, which again I'm not going to touch upon in this lecture, but it's essentially also these hippocampal cells, they show very prominent oscillatory properties, so you can look at them as oscillators. And so the idea was that a bunch of these oscillators sum up, which can result in these kind of waves, and then they will sum up in grid cells, which will result in this stable pattern. And then, this is experimental evidence or result. So here you see a sagittal brain section, which is like from this side. And this is entorhinal cortex, this is hippocampus. What we do know experimentally this is that going from top to bottom, so from dorsal to ventral direction, grid cells have different scales, from very small, and that's across animals, very, very reproducible. So from somewhat 30 centimeters, something like that, to larger scale, which actually go, we think, in a kind of discrete steps. So we can, again, predict quite nicely that usually within any rat or mouse, actually, even though the sizes are different, we will measure 30 centimeters grid somewhere here, and then going a bit more down, we will measure something which is 1.6 times larger, and then down 1.6 times larger, and so on and so forth. So it's kind of in the discrete steps. So what you can see now is essentially that we can sum them up, kind of like a Fourier transformation, in a Fourier-like transformation, to get from these different scales and different orientations, single bumps of activity, which essentially would be place cells. So that was the very prominent models of how you would go from grid cells to place cells, how you would generate grid cells, and so on and so forth. And it all went into kind of like a very nice story of how the system, navigational system at least, works. Except perhaps there was some, let's say, new findings which did not go extremely well yet, at least with these theories. So I should mention two of them done, but yes, some of them done in Norway still with by Mosers, who discovered grid cells, and by other people as well. So people looked at developing animals, so very, very young animals, which just leaving the nest, and they tried to prove this theory of going from grid cells to place cells by measuring, by looking which cell type appears first, which signal appears first. So what they expected to see that in a very young animal, we will see a lot of grid cells, place cells somewhat unstable, and then they should emerge. So that's not what happened. It happened the other way around. Place cells emerged when the animal was around P16, and this is the time when they leave the nest. And grid cells emerged 7 days later. So place cells emerged first, not grid cells. Another study was done in adults where, again, I'm not going to go into details, but you can manipulate another brain area and essentially very specifically get rid of grid cells, but not place cells. So it became clear that it's not that clean story. Another finding without disrupting system, all looking at very young animals, coming from our lab, where we show that grid cells, I already mentioned to you that they usually have a hexagonal symmetry as a kind of crystal structure, But actually what we saw is that it's not always the case, and it was a continuum of various symmetrical properties found in the cells which were more or less located where usually grid cells would be found. And that these deviations from the grids very much were triggered by an animal going from one shape of environment to another shape, which prompted us thinking about boundary conditions as important factors setting overall global grid cell symmetry. Which again, if you remember those two models that I showed you, none of them would be predicted any kind of sensitivity to boundary condition, at least in the form that they were presented back then. So how we try to look at that is to just see, not so much in a physiological sense, at that point at least, but more conceptually, how we can get a system which would be sensitive to the boundaries of environment and which would somehow recapitulate what we see experimentally. And so we decided to look at the grid as instead a collection of individual fields, so place fields, for instance, if you like. And you can think of them as a kind of place fields which would interact with each other. And they would interact with each other in a way that they would try to keep a distance from each other in like a particular optimal distance, which essentially would define the scale of that crystal. So if you imagine that you allow this system to, you have an infinite space and you have infinite number of circles interact in that way, you will very, very soon converge to a closest packing arrangement, which is essentially a hexagonal symmetry. And you can add boundary conditions to that infinite sheet. And in our case, it was a force, what we call force, which would be repelling these circles from the boundaries. And again, using or choosing specific interaction parameters, you could show that kind of system could easily converge from let me just try to play, from any arbitrary starting location to, this is like a convergence trajectory for each starting location, to another position, and that stable position for each one of them essentially would be formed in this hexagonal symmetry. So now going to more physiological domain, which again, it's very speculative. I'm not suggesting that it is true and it's now the most dominant theory or anything like that. But if you could imagine that every individual field, every circle could be looked at as input from competing place cells. And indeed, we do know that in this region CA1 that I showed you in the hippocampus, which actually does send strong projections to the medial entorhinal cortex. So I told you that entorhinal cortex projects to the hippocampus, but it also goes the other way around. So here you have a bunch of these individual place field representations, which anatomically do connect to each other, but via inhibition. So via cells, so not direct projections where one cell would excite, so make another cell fire, but actually it would excite a cell which would silence the other cell. So that's what we call via inhibition. So these place cells compete essentially via these inhibitory so-called neurons. And then you also have border signal actually also very closely located to CA1 region here in subiculum. So you can imagine border cell also compete via inhibition, and then whoever wins ends up projecting to grid cells. So in that sense, it was quite controversial, suggesting that, well, maybe there is a larger role for place cells to play in this whole formation of a grid. And importantly, what this model predicted, and at that time it was real prediction, not post-diction, because experiments were not done at the time, was that, okay, now that we have this framework of interacting place cells and boundaries and so on, we can look at any shape of the environment. So because usually in the lab, it just happens to be historically or whatever, people are looking at circles, squares as the simplest, let's say, shape, the simplest environment that the animal can be tested. So what we did was reasonably simple, I think. But we expected that it will tell us all about how grid cells are generated. So we just tested the same grids in square and in trapezoid, and we had our predictions as to what should happen to the symmetry of the grid. So this is a model, and this is what we got after we did experiments in squares and trapezoid. This is an example of just a single cell firing in a square and trapezoid, but actually very similar tendencies were seen throughout. And if you do simulations of that kind of interaction, the whole difficulty of this whole thing is that you would have a distribution. So it's not like you can't get hexagons in trapezoids, you can, but very often you will deviate from it, which will happen much less often in circles and squares. So that was, prediction, and that's what we also found very much so experimentally. So what we wanted to do further, we wanted to test it much more systematically. So instead of jumping, let's say from, metaphorically speaking, jumping from square to trapezoid, we actually chose another polygon, which we transformed to rectangle. So in this case, I showed you as if it's some kind of smooth transformation going from this left-handed trapezoid to rectangle, but actually we randomize shapes to prevent any kind of hysteresis or anything like that. And what you see in here, so sorry, just to reiterate what was the purpose of that experiment. So the purpose of that experiment was to see the nature, to look at the nature of transformation. And the question was very simple. Can we transform a grid on a single field basis or local field basis, or is it more global, what exactly it is, and so on and so forth? So that's number one question. And number two, in these experiments, we also recorded from place cells, including simultaneous recordings from grid cells and place cells, trying to see then whether we can connect what happens on the field level in place cells versus grid cells, and so on and so forth. So what you can see in here, hopefully, is that when we are changing this one so-called slanting wall, the majority or the fields on the right-hand side more or less stay untouched, undisturbed, preserve their position. Whereas fields on the left-hand side, and especially that field, very much moves with the wall. So that was already very interesting to us, and we tried to quantify exactly what was the range of this movement and so on and so forth. So, did I do something? It's a sign of ending a talk. Yes, okay, I will speed it up. So in a nutshell, again, I'm running out of time, but in a nutshell, what happens, so here you can see vector fields going from dashed environment, so here in left-hand side polygon, to another environment is shown in solid, of all the fields, average fields from all the cells combined. So in this case, we recorded from 346 grid cells. Not at the same time, it's different animals and so on and so forth. But when we combine all of them and look what happens in different positions, you can hopefully very quickly see that the fields very close to the slanting wall move a lot, and almost nothing happens on the right-hand side. And this is kind of showing the same thing, but quantifying it in terms of choosing what's significant movement, what not, and so on and so forth, and what directions they would be moving. Initially, I thought it would be horizontal, vertical because of the boundaries, but actually it's also perpendicular to one of the boundaries, which again makes sense. Anyway, so then we spent quite a bit of time trying to come up with a model which would explain these kind of movements. And you can think of it, well, at least in several ways. So this is another example of a grid, which was quite remarkable and kept me at least working for a while, because it was very deceiving in many ways. So you can see field number 1 here, and then you can see as if 2 fields appearing. So the first idea, looking at this striking example, was, oh, okay, the field split. One one field stayed locked to the west wall and the other one to the south wall and just kept a relative distance constant. So this is the schematic representation of that. You can see that, you can imagine that if each field would just try to keep its relative distance to the wall constant, to any of the wall, you can see that, well, of course, nothing would happen due to the north wall, east wall, because they don't change, but the west wall, this field would move with this wall, and so its new position would be here, and in the south, this very same field would move up, so very kind of similar to what we saw in this example. But kind of counterintuitively, in another way, how you can think about it is that the whole grid now, so not on individual fields level, the whole grid scaled together with the wall, so moved with the wall. So you can see here in the initial environment it looks like this, but let's say it scales, so it's anchored to the east wall, and then it just contracts. So in this bizarre situation, when our environment became smaller, we actually see more fields. And so that actually, again, I want to skip now a couple of slides, but it turned out that that kind of prediction or that kind of working hypothesis could much better recapitulate what we saw from one environment to the next. And just to show you that indeed with the border cells, as shown in this model here, they very much moved with the wall. So you can see that here it was active in the west wall, and here it moved with the slanting wall. So it's not unreasonable to believe that there would be this signal from the border, and if that somehow is coupled to the fields, they would move together or something like that. So this is just to show you what I just said, that if you just keep the relative distance of each field to the wall, and you would try to simulate what your pattern should look like, and compare to the pattern that you actually measured, your correlations would be not that high. So you would not perform better than if you didn't do anything. And you doing nothing on the west is also not good enough, because it's much worse than compared to the east hand side. Yeah, so it was not very good model. Another thing about it was that, or observation about it, was that this type of interaction would predict that the shift of the field would depend on the scale of the grid cell, and that again was not the case. So it didn't matter what the scale was; the shifts were pretty much constant. Then, but as I said, another model was that we keep the grid fixed to the stable side, so east side, and we just contracted it by, let's say, the same amount. And you can see that in that case, there would be almost no change if, let's say, the scaling was 10% or so. There would be almost no change on the right-hand side, but that change would become more and more prominent as the wave, if you like, keeps on traveling. So for expansion and contraction. Indeed, when we looked at the kind of changes in on average scale within those grids, we could see that there was quite prominent change on the order of 8 to 4%. But the question still for us remained in the context of our model, the one that I told you about the interacting fields, was whether that rescaling was uniform or non-uniform. And again, it makes a difference as to interpreting what's going on. So how we addressed this problem, we took, this is another example of the grid cell, grid field. So we compared one equal area of one environment to the area that would be most similar to it in another environment, and would then see whether it's expanded in terms of pattern, I mean, or contracted, and by how much. So like this, we would proceed like so. So you can see that here we already see the entire field exposed, and here it's just not fully exposed, and so it goes even more. So almost none appears here, but already the two fields appeared here, and so on and so forth. So we were interested to see what kind, it's clear there was a rescaling, but what was the profile of that rescaling? And again, not to complicate things and rush through the slides, this is what these profiles would look like, and this is simulated profiles of a homogeneous rescaling. And what we saw is that at least in some types of transformation, that rescaling was not homogeneous. But again, because of such a limited pattern exposed, even though they were large environments, it's still very limited for that type of analysis. What we are trying to do now is to record from much larger environments, largest we can get, essentially, to expose even more of that crystal, if you like, and so on and so forth. So again, now going forward from grid cells to place cells, you can see that place cells actually showed very similar tendency to grid cells. So you can see that field, for instance, here, which was close to the slanting walls, literally moved with it up and then disappeared. And something further away stayed fixed. And this is, again, the average vector field for all the place cells that we recorded. So the same tendency or similar tendency. But interestingly, place cells always overshoot grid cell shift. So they are not one-to-one. And even in situations where we co-recorded them together. So in this case, you can see grid cells black and blue cells, blue arrows corresponding to place shifts. So they do show the same tendency, but they are not correlated significantly enough to say that it was the same type of movement. And there could be different reasons for that. One is that there is no one-to-one correspondence between the two types, or even though they're simultaneously recorded, we don't have any information about the actual cell interconnectivity. So what we are trying to do now in the lab is to actually look at activity of synaptically connected, directly synaptically connected cells and see how well they are coupled to resolve that question. What we also did, we looked at how this movement was correlated among different co-recorded grid cells. And here we used a very new technique developed at UCL in partnership with Janelia in the United States and Alan Brain and so on. And actually, Marius Bouja led that project, and he contributed to that study. So in this project, we were able to record really quite record numbers of simultaneous number of grid cells, so in this case, I showed 24, but we were able to ramp it up to 50, just to tell you, before that technique appeared, the average number of recordings would be 5 to 7 cells per rat. Sometimes it's higher, sometimes it's lower, sometimes it's 3 cells. It's quite difficult to find them. But with this technique, we actually had a quite major breakthrough. So we were able to look how coordinated these transformations are, and essentially the answer was that they were extremely coordinated across different scales and so on and so forth. So just, again, reiterating, using the very same analysis for collocated grid cells and place cells, they were not significantly coupled. So these guys are very coupled, place cells, grid cells, not that coupled. And we'll use that information also for the first time from the real data, and this is Marius's work, to essentially, again, due to the time limitations, I'm not going to go into details, but to show that we can use this cell activity to still correctly decode animal positions, even from these distorted, transformed grids and so on and so forth. So in principle, they can be used for positional decoding, because from our trapezoid, let's say, experiment, it was not clear at all, and we speculated maybe it's not a metric for measuring distances and so on and so forth. So it still could be in principle, but what that would mean is that either there is another system which corrects for that in the brain, or animal gets positioned wrongly in a transformed environment. So we started testing it. This is not published and just literally beginning Essentially, we started testing it in a kind of behaving animal doing a task. So previously, when I showed you all these cell recordings, the patterns in trapezoids, squares, circles, and so on, that was using so-called tetrad recording, so electrodes stuck or implanted in animals' brain when they more or less freely foraged for food in that enclosure. They didn't do really any other task then, nothing special about it. But now we actually wanted to ask animal, can you find food in a particular location? And we used two pieces of information, which was published in 2004. Where these scientists claimed that, well, two important things for us at least, is that animal can learn to dig for food in the center of the environment, so they can make internal estimation of what is the center of any enclosure. And importantly, they can generalize that knowledge, so they can kind of know that, oh, food should be in the center. Wherever I go, food is in the center. Circle, square, trapezoid is in the center. So they can generalize it to then look for food. And so then they would be very interesting to see how much the center, and we should be able to predict systematically how much they should deviate in various enclosures and related to the cell activity and so on and so forth. But as a first instance, it was even interesting, how do they even do it? And based on my own experiments in rats, so this is our own study. Now this is all done in pitch dark because we only wanted to force the animal to use geometrical information and nothing else, so no visual, no nothing. We also controlled for tactile, smell information, and so on. So I was wondering how they even sample the geometric properties of the environment. So this is the first minute or so of the rat running around in a square. So you can see that even with this 1 minute, animal knows in principle the shape of the enclosure. And this is an example of one rat, but actually it applies to more or less any rat, actually, on average. So then what I thought that it would be super interesting if you put the animal in any kind of geometrical enclosure, as is done in this experiment, and they indeed would go a perimeter, form some sort of shape representation, and then go to the middle, for example. Well, you will see in a second, at least from the pilot data, that's not what they do. So this is a trained rat, which also in the dark, this is pitch dark, which looks for food in the center, was trained to look for food in the center. So the center is shown in orange, but it's just for our purposes. There is no orange mark or anything like that. And you can see where he starts digging. In the complete dark, there is no food actually placed there, so there is no smell information or anything like that. And he quite accurately finds, I would say, a location of the reward, estimated reward location. But he does it in a very different way. He essentially measured two local corners of this environment and then made its way to the goal, to the reward location. And now in this final movie, it's a rectangle environment which he never visited before. And he was trained on that square, and now it's his task to dig or not to dig. And you can see that Wade would start digging. So it makes quite a big mistake. So the real center, of course, would be green, but then this would be actually half of that center. And again, you could see that it measured one local corner, and it proceeded here. It's probably in expectation to find another, and it would turn here, but there was no. It's proceeded here, and so on. So what we were trying to do, basically, is to relate the perception of where something should be located, especially if it concerns the middle, and try to relate it to place cell and grid cell activity I would not say it's the first study trying to do it, but there are still very few studies trying to actually infer how animals are using these cells, mostly because it's very, very difficult to ask animal to report what exactly they're up to. Okay, so in summary, thank you very much for your attention, and I'm sorry to be quite late, but hopefully the take-home message is that hippocampus is absolutely important, plays a key role in episodic memory and navigation. So it's our GPS/episodic system. And there are a lot of, I think we understand a lot about how navigational system works, not so much episodic memory system, actually, but it's another talk. But still, there is a lot of progress to be made as to, especially in the domain of how this whole ensemble of various types of spatial cells interact to generate the actual firing properties. And then, very importantly, how they are used in navigation perceiving space, because I think at least in this field, okay, there is, of course, this fundamental question about how we perceive space and so on, but there is also, I think, idea of using this spatial framework, hippocampal spatial framework, entorhinal spatial framework, to understand how our memories work. So I think that's the kind of the final aim of, or the final game, let's say, of this research. Okay, thank you very much.
Thank you, Julija, for an impressive talk, and we have a couple of minutes for questions. Should I come with a, do you have another, or do I have to come with this one? Sorry.
Doesn't matter.
But for people who are on the other side of online, they won't hear your question.
Oh, maybe you're making Thanks, Julija, very impressive. I just wanted to ask a couple of questions. So first of all, you showed that there is a lot of redundancy in the place cells. Why do you think there is redundancy? Is it just robustness? And maybe a related question. So we know from our own experience, so when we enter a simple geometrical space, there is no arousal, and when geometrical space is more complex, there is certain level of arousal that disappears with time. So, and because we form memories and we navigate space differently. So is there a fundamental difference between, let's say, humans that are able to form memories and rats? So do you think that redundancy is perhaps diminished once the memories are formed? Or is it too difficult to answer so far?
Okay, so I will should I repeat? He had a microphone. So, okay, so the first question about the redundancy. So I think, yes, the simple answer probably is because of the robustness of the system, it would be very sad, I would say, and dangerous if we had just one cell reserved for one place and something happens to that cell, and we know it does happen. But to answer it, well, experimentally, when somebody or animal enters a new room, actually more cells become active. So redundancy is higher in that sense. And when you get more familiar with the room, a lot of cells are so-called being pruned into a stable cell kind of ensemble. But even that is a bit more complicated. So the number of cells, so I mean the number of how many cells are active does stabilize, but actually they keep on changing. The ensemble keeps on changing. So if you can record for 30, 40, 50 days, whatnot, next day you come back, let's say 90% of yesterday's cells would be active, but 10% other cells kick in and 10% would disappear. And so it goes. But if the cell from day one re-enters at day 15, let's say, it re-enters in its old position, whatever that means. So that information is preserved. It does disappear. There is a dynamics to it, but right now, again, it would be a separate talk. There is research going on in this, and that's much more memory related now, and people speculate it's episodic memory and so on, whatever that means. Because actually when people speak about it, when they, in the context of animals, because it's very, very difficult to ask animal anything about episodic memory, but more or less anything that there is a temporal changes, that would be called episodic memories in animals as it stands now. So some information is preserved, as I said, it's very dynamic in terms of number of cells. It goes down stably, the number of cells. in the novel environments. So that's about redundancy. And as I said, I think right now we think the redundancy exists because of the robustness of the system, but it could be that it's because different other systems, neocortex and so on, are using different cells which represent the same place, but used by different regions. And again, there could be some point to it because even though they're all place cells, they actually are quite heterogeneous as to what type of information they prefer. So again, it's a separate talk maybe. So that's one question. Another question about complexity of environments and arousal and so on and so forth. It's actually an interesting point. Are they less aroused in primitive shapes? I'm not sure, maybe. Just saying from my own experience, so all these patterns that I showed and analysis and so on, that was all done independent of shape, was all done for highly familiar environments. And the reason being is that indeed there is a novelty so-called effect on these cells. So in the new environments, both place cells and grid cells become larger, messier, and so on. They converge to those beautiful whatever crystals. And what happens is that the convergence in more complex shapes, so trapezoids, for instance, which are comparable area size to squares, takes longer. So time-wise, it's more difficult, if you like, to cope with the more complex environments on average. It's animal dependent. Some animals are better than others, but on average, that's the case. Again, how it's related to arousal, I don't think it has been asked. I mean, I could maybe speculate that there are lots of emotion-related systems directly connected to the hippocampus, so they could play a role in that, but that would be pure speculation. It was not asked. Yeah. And there are actually, so just to mention, to end this answer, is that there is now a study going on, follow-up, essentially from the study we did in square trapezoids in, I believe, Trondheim, actually, so not by Mozart, but another group, in humans. So they are trying actually to replicate it or to at least address it in humans using other techniques, fMRI essentially, and VR, and so on. So we'll see.
Any other questions?
Yes, how large the area covered by the grid cells can be in open environments, like in natural conditions?
Yeah, so how large by grid cell? I think infinite. That would be a short answer. If you're asking how large the scale could be of a grid, what's the largest scale? That right now, at least based on kind of, remember I told you that it's a fixed ratio, how they go, and how much people could record and how long is the whole structure. People think it should be 10 to 11 modules, so you can 30 centimeters times 1.6 power to 10. So that's kind of the power to distinguish. But as I said, it depends what your question was. If you're asking how much they would span, I think infinitely, more or less. But the largest probably environment that I know of recorded, because of course we are limited by experimental conditions, right? And space is a premium in London and other places. So I think it was done on a linear track, and the linear track spanned 18 meters or so, and that could be easily covered. Yeah.
So thank you. I am signaled that we have to finish, unfortunately, with this part to go to the short talk session. So again, thank you, Julija. And